The overall goal of this 3D-PAGE procedure is to separate protein and ribonucleoprotein complexes from Brassica napus phloem sap, coupled with the identification of the respective nucleic acid and protein components.
This method can help to answer key questions in biochemistry and plant physiology. For example, you can analyze the composition of phloem complexes under stress. The main advantage of the technique is that pure phloem sap can be collected, and nucleic acids as well as protein components can be analyzed simultaneously.
Though this method can provide insights into protein-protein and protein-nucleic acid interactions within the phloem system of oilseed rape, it can also be applied to phloem analysis of other plants like cucurbits, lupines and yucca.
We first had the idea for this method when we tried to identify protein and RNA interaction partners for specific phloem protein.
First, puncture the inflorescence stem of an 8-week-old bee napus plant several times using a 0.8 mm hypodermic needle. Puncture several other inflorescence stems on the same plant and other plants to obtain enough phloem sap. Wipe off the first drop from each injured site with filter paper. After removing the first drops, use a pipette to collect the remaining drops and transfer them to a pre-chilled conical 1.5 milliliter reaction tube. Then, store the collected exudate at -20 degrees Celsius using an ice-free cooling system. Continue collecting drops until no further drop formation is